Hello, I'm François Réard, NanoSIMS product manager at Chemica in Gennevilliers, near Paris in France. So this webinar is the first of a series of two focused on trace element mapping in cell biology. And there will be two parts. First, trace element mapping in tissue sections, and then metal-based drug mapping in cultured cells in the field of oncology. This slide describing the instrumentation is explained in more detail in the first NanoSIMS instrumental webinar, available on Chemica website. In short, a primary beam of reactive ions, cesium or oxygen, is focused and bombards a sample placed under high vacuum. The focused ion beam is scanned pixel by pixel, and for each pixel, atoms and molecules are sputtered, and the spontaneously ionized secondary atomic ions are extracted with a strong electrical field and injected into a magnetic sector mass spectrometer. This spectrometer is equipped with 7 mobile detectors for 7 selectable masses. And if we change the position of the detector and the magnetic field, we can access masses between hydrogen up to plutonium or higher. The analyzer is working at high mass resolving power, removing most possible mass interference. Then the analysis is of destructive type, so it consumes for each image a few nanometers. So 3D information is accessible with a depth resolution of roughly 10, 15, 20 nanometers. So lateral resolution down to 50 nanometers. It can be adjusted between 50 nanometers and 1 micron, 2 micron. And depth resolution roughly between 10 and 20 nanometers. Sample preparation. So it is usually extremely similar to transmission electron microscopy sample prep. Basically, the analysis is done under high vacuum, so any sample must be dehydrated. So no analysis on live cells is possible. Sample must be flat, so generally we work with thin sections, and conductive, so if the sample is not conductive, like for bones, for example, we use a metal coating. First, sample must be fixated, whether by fast freezing or chemical fixation. Then ice must be removed, so whether by drying or substitution with alcohol and resin. Then a microtoming can be applied to obtain a thin section, whether we can image often the same section with SEM, electron microscopy, or TEM, or we use serial sections, especially for optical microscopies that require staining. They are done on serial section. First, for navigation over large sample, we use optical microscope inside the instrument with a resolution of 1.5 micron, roughly. Then, for histology, for recognizing the cells and the structure, we use CN, so nitrogen for the protein, phosphorus for the nucleic acid, and sulfur as images, giving us an excellent contrast without any chemical staining. Like, for example, in these cells, where we detect, using cesium, we detect negative ion. If we were using oxygen primary beam and detecting positive ion, we would use calcium, potassium, or sodium to have access to intracellular structures. So the membrane, the cells, the nucleus, nucleoli. And then it's possible, after this identification, to start mapping trace elements. We can make also another use of elements by using specific isotopes of given elements. So in this example, these researchers in this paper from Tang in 2014, the researchers are studying metabolic pathways of selenium. So they use aortic endothelial cells that are incubated with different sources of selenium. And these sources are tagged with specific isotope of one element, selenium. So for example, selenomethionine is labeled with the isotope of mass 77 of selenium among 5 other natural isotopes. And we can map on the lower row, on the right side, the distribution of 77 selenium over the major one, 80. And you see the distribution of this selenomethionine molecule. On the left side of the lower row, you can see cells that have been duplicating recently and have incorporated thymidine, but thymidine labeled with nitrogen 15. And what you see in red are in the nucleus and inside the DNA that have recently incorporated thymidine nitrogen 15. So we can make use of elements for localization, specific elements for localization, then trace element for quantitative measurement and distribution, and also use specific isotope of one element. These slides will illustrate the great power of combining different imaging and analysis techniques at different scales, and it will be based on a paper by Christopher Chang in Metallomics in 2018, dealing with copper in a retina photoreceptors in the zebrafish. So copper is essential for eukaryotic life, and mutation in a protein, ATP7A, contribute to a spectrum of diseases, including Menkes disease. So fundamental understanding of this, of the impact of this mutation on in vivo copper distribution is interesting. Here, different techniques, it was optical microscopy, confocal optical microscopy, laser ablation ICP-MS, transmission microscopy, and NanoSIMS, were used to study and understand copper level, altered copper level in zebrafish model of Menkes disease. So there is in this allele a point mutation in the protein that causes impaired copper export. So in this first image, you can see in the upper row an optical microscope image with nuclear staining, revealing the different layers of this cross section of retina. And what you see from left to right, the ganglion cell layer, the inner plexiform layer, the inner nuclear layer, the outer plexiform layer, and the outer nuclear layer. And finally, the retinal pigmented epithelium. And what is imaged here in the second and third row is copper signal and phosphorus image. And in the lower row, you can see a superimposition of copper in blue and phosphorus in red. And first, the copper has been quantified using laser ablation ICP-MS. So the NanoSIMS images here are displayed in a quantified scale in ppm, part per million concentration of copper. And what you see copper in the outer nuclear layer with puncta enriched in copper. So this is on the wild type embryo types. And when you compare with similar image recorded and embryos that have this problem of export of copper, you see that the main difference is the level of the concentration of copper in this small puncta, in this outer nuclear layer. This slide shows the interest to correlate, in the second step, the chemical mapping of NanoSIMS with the high resolution of TEM, transmission microscopy. So on the zoom image on the right side, the mega mitochondria have been colored in blue and correlate very well with the region showing in the NanoSIMS the copper enrichment. So the comments is that imaging of the neuroretina with NanoSIMS is combined with electron and confocal microscopies, identify the megamitochondria of photoreceptors as loci of copper accumulation in wild type zebrafish. So it's then you have sensitivity with the NanoSIMS and resolution, ultimate resolution, nanometer resolution with the TEM. So let's use now some results provided by Dr. Charles from the IPREM Pau in France. These are examples of application of the NanoSIMS in plant science to localized metals. Metals in the study of photosynthesis, and photosynthesis is done in leaves at the level of thylakoid membrane in the chloroplast. So, during the photosynthesis, many chemical reactions take place in the thylakoid membrane level, and they are involving metals such as iron, copper, zinc, etc. So the question is detection of these trace metals possible with the NanoSIMS at a resolution of 50-100 nanometer? The answer is yes. The detection of manganese, iron, copper is possible at the physiological level in plant cells. And this is true for most metals. This is a characteristic of the NanoSIMS, so good sensitivity together with 100 nanometer resolution, except for a few elements that are not very sensitive in SIMS in general. Zinc and cadmium cannot be detected at ppm level at this special resolution. Rare gases are not detectable, and mercury is not detectable in the NanoSIMS. This second series of images, taken from a paper from Mr. Malherbe in Analytical Chemistry 2016, show in fact the accumulation of 30 planes through the section, so going in depth through the cell section, and revealing membranes, enriched or not, in different metallic elements, and tiny granules at different depths, enriched with different metallic elements. So, this also illustrates one difference with standard TEM imaging, which integrate the signal over the whole sample thickness. Whereas in the NanoSIMS, each image represents a small depth of a few nanometers, and the stack of image can go down to micron thickness. So it allows 2D maps, 1D depth profilings, or also a pseudo 3D reconstruction with a depth resolution typically in biological tissue of 10 to 20 nanometers. Let's now illustrate the capabilities of the NanoSIMS in pharmacology and cancer research. For this, we'll use an article by Ronald Lee in Metallomics 2017. In this article, ovarian cancer cells were cultured, then fixed by high pressure freezing, then embedded with resin, and finally microtomed to serial sections. So thicker section for light microscopy and for NanoSIMS, and thinner section for TEM. So here you see untreated sample of a cell line sensitive to cisplatin, the A2780. So we see the platinum level normalized by the carbon of the section of sample, and it can be compared to a cell, same line, sensitive to cisplatin that has been treated this time with cisplatin, and immediately we see variation of intensity in the color scale of the platinum inside the cell. It can be compared to cells, another cell line, the A2780CR, so cell resistant to cisplatin, where we can clearly see a much lower level of cisplatin or platinum inside the cells. So this illustrates these images have a resolution, lateral resolution of, let's say, 100 nanometer spatial resolution. So we have enough sensitivity to see differences. In order to get a better spatial resolution, combining with transmission microscopy is extremely useful. It allows to identify subcellular organelles. So in this exact example, images are shown at the same magnification and position between phosphorus, sulfur, and platinum obtained with NanoSIMS, and you see also correlated the TEM images. We can see in the platinum some hotspots in platinum, and one can review then inside the TEM, the high resolution TEM, and one, for example, in the lower image, can see that you have an increase of platinum into the autophagosome. It's exactly correlated with the hotspot in platinum. Now we can see a comparison with a resistant line to platinum, to cisplatin, treated, and we see different accumulation of platinum here at a much lower level, but still some discrete slides. So, in short, NanoSIMS brings sensitivity at 50, 100 nanometer, enough to localize, highlight differences of concentration, and TEM can identify precisely the organelles. We see here a reduced accumulation of platinum in cells with acquired resistance to cisplatin, and it was the first direct observation of platinum accumulation in mitochondria and autophagosomes. And it highlights the importance of nucleolus-based targets for cisplatin. So, how do we extract quantitative data from images? In fact, after the acquisition, the pixel intensities are summed within region of interest, and we can measure quantitatively like this by adding pixel from region of interest, ROIs, that can be hand-drawn, like here, or automatically defined, for example, with chemical software, WinImage. And like this, we can obtain measurements from specific compartments of individual cells. Can distinguish here, for example, cytoplasm, nucleolus, and we can compare inside a cell line, but also between different cell lines, here in this second set of images, from cells resistant to cisplatin. So we can see quantitatively, we can compare enrichment localization of platinum. We can see variation within individual cells of the same line, and also here in this case, a reduced accumulation of platinum in cells with acquired resistance to cisplatin. Let's now use the article by Ronald Lee in Chemical Communications in 2015 to switch to another element used in oncology, ruthenium. So here, images of cultured human ovarian cells were recorded on microtome sections of resin-embedded samples. And the strategy for NanoSIMS analysis was here to synthesize the ruthenium-based molecule with the toluene ring labeled carbon-13 and the phosphine PTA ligand labeled nitrogen-15. So with the NanoSIMS mass spectrometer, it is then possible to image in parallel the metal center through ruthenium ion, the toluene ring through carbon-13 over carbon-12 enrichment, and the PTA ligand through nitrogen-15 over nitrogen-14 enrichment. Let's directly jump to some of the conclusion of this article. And this conclusion, first, the phosphine ligand remains coordinated to the ruthenium ion, whereas the arene detaches. And the complex localizes mainly on cell membrane or at the interface between cells. And the main output concerning the NanoSIMS usefulness here is that the stability, liability of the ligands coordinated to the metal center can be probed through isotopic labeling. So let's now see the measurements leading to these conclusions. The control cells, so line cell A27ATCR, on untreated cells, the cyanide, phosphorus, and sulfur images allow to recognize the different compartments. The carbon-13 enrichment and the nitrogen-15 enrichment image are quite flat around the terrestrial level. One can notice some slight carbon-13 ratio difference between cell and resin. And finally, at the bottom right, ruthenium image displays a very weak background signal. Now, the treated cells. So the cells that were treated with the ruthenium drug show first a very similar carbon-13 image compared to the control, but different nitrogen-15 enrichment, so the PTA ligand, and the ruthenium image. So one can extract a line scan from the white line, and conclusion one, there is a clear ruthenium and nitrogen-15 co-enrichment observed on cell membrane and at the interface between cells. The nitrogen-15 labeled PTA ligand remain coordinated to the ruthenium center after 24 hours. And second observation, taken from the line scan on the carbon-13 enrichment, there is no significant carbon-13 enrichment. So that points toward the toluene ligand detachment from the complex and dilution over the sample. one can display the images superimposed in red and green colors. So phosphorus is useful to recognize the cell compartments, nucleus, nucleolus, cytoplasm, nuclear membrane, and cell membrane. And a second remark is that ruthenium was analyzed here in negative mode. So the ionization in this mode is not very high. So the image acquisition is very long, maybe nearly a full day. In the future, it would be possible to record ruthenium plus under O- bombardment to get a better ionization. But even with these low yield conditions, the instrument is stable enough to keep the intracellular lateral resolution. So in overall conclusion, through this observation, the NanoSIMS allows probing the stability and liability of the ligand coordinated to the metal centers via isotopic labeling and, of course, mapping of metal, here the ruthenium, at low concentration. In the same field of oncology, let's switch from platinum to gold and use this recent article by Ka-Chung Tong in PNAS in January 2020, Anticancer Gold-Based Drug Development. So here, the researchers synthesized 3 molecules based on gold. So gold meso 9, gold TPP, gold OEP with different structures and adducts. And ovarian cells were treated with the first molecule, and we can image clearly with CN image and sulfur image the nucleus, the nuclei, and we image the gold incorporation. And correlating with high-resolution SEM, one can see that the gold is enriched in mitochondria. And when comparing with different molecules, so gold TPP on the ovarian cells, one can easily see with the NanoSIMS the different gold internalization between the molecules, because of the high sensitivity of the NanoSIMS on gold at typically between 50 and 100 nanometer resolution. Gold meso 9, exhibiting effective antitumor activity in mice, is localized outside the nucleus. So now we see the 2 images on the left, gold distribution using gold meso 9, and as a small aggregates in the cytosol, and the middle image shows sulfur over nitrogen image, and the third image shows the superimposition of the 3 elements. So there is a very clear co-localization of gold with these sulfur-rich proteins. And with the gold TPP, there is only a diffuse gold signal in the cytosol. No correlation. Then high-resolution SEM image, electron microscopy image, could be done after that, showing electron-dense aggregates in degraded mitochondria. On the right side, you see intact mitochondria that are very different from the observed one after the interaction of the gold meso 9. So in conclusion, the combination of NanoSIMS and electron microscopy could reveal a tight association between the gold and sulfur-rich proteins in the form of aggregates in the cytosol and disrupted mitochondria. So this study has uncovered a gold-3 induced ligand scaffold reactivity for thiols targeting that can be exploited for anti-cancer applications. So the mechanism, the periphery of coordinate ligands is activated by the electrophilic metal center to selectively form covalent adducts with cysteine thiols of cancer-associated protein. So, in conclusion, the NanoSIMS 50 is an original ion microprobe allowing mapping of trace elements inside cells and tissues with a lateral resolution down to 15 nanometers. The measurements can be quantitative with the use of standards. Second point, the analysis is performed under vacuum, so on dehydrated cells or tissues. Sample preparation is very similar to SEM or TEM, electron microscopies, that are often used as correlative imaging to resolve nanometer scale intracellular structures. So the unique couple sensitivity spatial resolution of the NanoSIMS permits to map elements or molecules containing specific elements inside cells that are inaccessible by other imaging techniques by lack of selectivity or sensitivity. An Excel spreadsheet compiling more than 1,000 published scientific articles containing NanoSIMS data is available on Chemica website, so you can download it and filter the articles by keywords, subject, journals, country, etc. Thank you for your attention.